It's my pleasure to introduce Sheng Wang, who got his bachelor's degree at Peking University and his PhD at the University of Illinois Urbana-Champaign before taking a postdoc position for the last few years at Stanford. He joined the University of Washington as an assistant professor and is leading a group working on open world biomedical discovery. Today, obviously, he's talking about learning for never before seen biomedicine, and I'm excited to hear about some novel machine learning methods and ontological embedding techniques, as well as a multiscale biomedical knowledge graph, which sounds a lot like what we are trying to do in Semantic Scholar. But in addition to analyzing millions of papers, he's also managed to incorporate scientific experimental associations. So very excited. Thank you, and please take it away.
Great. Thank you for the introduction. I'm very excited to speak at AI2 today. I'm going to talk about a very important problem in modern biomedicine. So machine learning has shown promising results in biomedicine when we have enough training samples. But what if we don't have enough training data, or in the extreme case, we don't have any training data? For example, predicting side effects of a new drug or classifying patients to a new disease like COVID-19. I named this kind of problem never before seen class. It is a label class, like a disease or a drug, and we have never seen any training samples belonging to this class. For example, here we have 5 different training patient samples who are classified into 2 different diseases. We also have 3 other diseases, SARS, MERS, COVID-19, and we have never seen any training sample belonging to these 3 classes. They are the never before seen classes, and they will be the focus of my talk. So we will see some test samples, and they might belong to any of these 5 diseases. Now, the question we want to ask is, how can we classify a test sample into a never before seen class? This problem setting is similar to generalized zero-shot learning, but I'm going to talk about specific challenges in biomedicine later. Well, let's also talk about the solution later. Let's first look at what kind of biomedical problems involve never before seen classes. In fact, this patient disease classification problem is just the tip of the iceberg. There are many other important problems that involve never before seen classes. For example, we still don't know the mechanism of many new diseases and rare diseases. This is because we have only seen very few patients of these diseases. Side effects of new drug is another example. Drug side effect is the major reason why many new drugs are taken off the market. We cannot predict side effects of a new drug because we have never seen any patients taking these new drugs, except those very few in the clinical trial. So problems caused by never before seen classes are pervasive. Especially in this big data age, we are often collecting and integrating data from different species, tissues, and populations. So when we do such a joint analysis of all these data sets, we will inevitably see lots of never before seen classes. And if we are studying new drug, new disease, new cohort, we also need to consider such classes. So now you can see that there are so many problems caused by never before seen classes. These problems seem to be very different and are currently tackled by different biologists and doctors. But I believe that addressing this challenge might be the key and the general solution to all these problems. Just like how deep learning has become a general solution for many biomedicine problems when we have enough training samples, we want to find another general solution, a computational solution for biomedical problems with no training samples. Let's go back to this example again to see 3 specific computational challenges here. Because we have never seen any training samples for those classes, we need to have some extra information. What we often have are class attributes. So in this patient disease classification problem, class attributes could be symptoms. Although we have never seen patients belonging to those diseases in this data set, other sources might have such patients and provide us with some class attributes. But why don't we just simply use class attributes to classify patients? This is because these class attributes could be very noisy, especially for new classes. If we only diagnose based on these class attributes, we could make a very significant number of misdiagnoses. For example, at the beginning of COVID-19 outbreak in December, the first few patients are misdiagnosed as SARS. So the first challenge here is to develop representation learning method to obtain high quality class representation. Okay, now let's assume we have addressed this challenge and we have high quality class representations. The second challenge is how to do the classification. The problem setting is that we have training sample for some classes and we have class representation for all classes. How can we use these class representation to transfer labels from these seen classes to never before seen classes? Okay, let's assume again we have addressed this classification problem. Now we can accurately classify a test sample into a never before seen class. The third challenge is how to interpret our prediction. Interpretation is very important in biomedicine. Interpreting those new classes is even more important because our knowledge about these classes is very limited. Just like we still don't have a good interpretation for COVID-19 prediction. All of my previous research focused on addressing these three challenges in the never before seen biomedicine framework. For example, one contribution I have made is to develop representation learning methods to embed different biomedical structures into class embedding space. Using these class embeddings, I have addressed important prediction problems in biomedicine like cell type prediction and protein function prediction. Then we have developed an interpretable model to interpret this prediction. So model interpretation is a very important problem in biomedicine. We integrate scientific papers and biological databases to build a knowledge graph for interpretation. These three steps together build up a very exciting framework from no labels to new knowledge. And it also provides us a general computational solution to different biomedical problems. Our method has been used in real-world biomedicine applications. We use them as the building blocks for different applications in different areas. We then collaborate with different biologists and fine-tune our method for their tasks according to the specific problem settings. This results in a very fruitful collaboration. For example, our representation method helps doctors at Mayo Clinic find the mechanisms of more than 400 cancer drugs. Our classification pipeline helps biologists in the Chen Zuckerberg BioHub accelerate their prediction pipeline from 3 months to only 20 minutes. We are currently trying to find the rare disease genetic factors for California newborn screening program. Our interpretation method and knowledge graph help scientists at NIH to understand their biological discovery. Today, I'm going to talk about these three parts: representation, classification, and interpretation in never before seen biomedicine. So let's start from the representation part. The reason why we need to do representation is essentially to help classification. Then why is classifying samples into new classes difficult? Let's look at an example here. Those gray ones are test samples. The remaining ones are the training samples. Here is the gold standard. Green and purple are the never before seen classes. So samples belonging to these classes only show up in the test set and has never been seen in training set. If a sample belongs to a never before seen class, we want to classify it into the specific class. So if a sample shall be green, but we classify it as purple, the prediction is still incorrect. Here is a prediction of a classical machine learning method, k nearest neighbor. You can see that KNN incorrectly predict the classes of any sample belonging to a never before seen class. This is not just a problem for k nearest neighbor. In fact, any machine learning method cannot make correct prediction by only using these features. Then how can we address this problem? The solution to this problem is to use zero-shot learning. And zero-shot learning can classify samples into never before seen classes if we have class attributes for these classes. Zero-shot learning has been used to classify images into new image classes. It is relatively easy to collect class attributes for image classes. This is because these image classes are often classes that we humans are familiar with, like animals or fruits. So we can often find high quality class attributes somewhere. But in biomedicine, the story is totally different. In most cases, we simply don't have any class attributes. This is a very interesting dilemma in biomedicine. So zero-shot learning has good performance when we have class attributes, but these classes are often old knowledge and less interesting. However, for new knowledge, like a new drug or a new disease, which is more interesting, zero-shot learning cannot do well because we don't have such class attributes. Like we don't have any class attributes for COVID-19 at the beginning. And biomedicine is all about discovery in new disease, new drug, and new cohorts. So how can we solve this problem? Our contribution is to use the biological class hierarchy to help us. So class hierarchy is a structured data that biologists use to document their knowledge. In biology, we call them ontology. You can think it as a two-dimensional binary adjacency matrix. Each node is a biomedical concept. Each edge connects related biomedical concepts. And even using this hierarchy is very challenging. The hierarchy is high-dimensional. There could be more than 20,000 nodes. It is also incomplete and noisy. The curation process could introduce many missing or incorrect edges. So we don't have class attributes, and all we have are these high-dimensional, noisy, and incomplete class hierarchy. Now the question is, how can we convert them into high-quality class representations? Here is the problem definition for this question. The input is a hierarchy. The output is an embedding vector for each class in the hierarchy. The prerequisite of our method is that these never before seen classes must present in this class hierarchy. So later I will talk about another solution where we don't have those never before seen classes in the hierarchy. How can we use literature to augment this kind of hierarchy? Right now, the goal here is to represent each node in this class hierarchy as a low-dimensional vector, such that topologically similar nodes will be embedded close together. So which two nodes are topologically similar given this hierarchy? A key observation is that this hierarchy does not follow the 6 degree of separation theory. Two leaf nodes on hierarchy could have a much larger distance than 6 degrees. So we need to explicitly model this kind of global structure. We use the high order proximity to calculate topological similarity here. So high order proximity can capture global structure of the hierarchy. It is different from low order proximity, like first order or second order, which only capture the local structure of the hierarchy. For example, for this hierarchy, we want to know which node is similar to the red node. If we use first order proximity, then two nodes are similar if they are direct neighbors. For example, this blue dashed circle is similar to the red node, but in this case, we only utilize very limited information on this hierarchy for this red node. That is only the direct neighbors. So how about we use the second order proximity? Second order proximity means that two nodes are similar if they share many neighboring nodes. For example, this blue dashed circle is similar to this red node using second order proximity because one of their neighbors is the same. So you can see that for each node, second order proximity utilizes a bit more information on the graph, not only direct neighbors, but also neighbor's neighbor. So why don't we just use nth order proximity? Here, n is the total number of nodes in the graph. In this way, we can capture the whole graph structure. Because class hierarchy could be incomplete and noisy, we want to capture the global structure to mitigate the noise. Using nth order proximity, any two nodes are considered as similar if their distance to all other nodes are similar. Let's look at the math to see how we implement high order proximity. The goal is to calculate a similarity score between every two nodes given this hierarchy. Here we use the diffusion model random walk with restart to calculate the similarity score. In each iteration, we will either randomly walk to a neighbor or restart from the original node. And after the diffusion is converged, we will get the equilibrium distribution starting from each node. And this equilibrium distribution models the distance from this node to all other nodes on the graph. So in this way, two nodes are topologically similar if their random walk equilibrium distribution on this graph is similar. However, these distributions are still high dimensional. We need to reduce their dimensionality. What we can do here is to jointly decompose the equilibrium distribution S of all nodes. We want to find an embedding vector X and a context vector U for each node so that the observed S is close to the estimated S hat. And Xi will be used as the class embedding for class I. So our representation can be used in both supervised learning and unsupervised learning. I'm going to talk more about application supervised learning later when I finish the classification part of this talk. Here, let's look at an application of our method in the unsupervised setting. So the goal here is to cluster cancer patients into different groups so that we can find personalized cancer treatment for each group. The feature is the genomics and the mutation data for each patient. We use our method to embed 6,000 cancer patients for patient survival clustering. Here, each data point is a cancer type with about hundreds of cancer patients. This figure shows that our method is much better than the one without using high order proximity. And this figure shows that our method is much better than the one without doing dimensionality reduction on those equilibrium distribution. So these results together show that our embedding enable personalized cancer treatment by accurately classing patients into different groups. We also have a few follow-up methods extending our embedding method so that it can be broadly applied to different problem settings. I'm going to briefly introduce 3 extensions. First, if we have multiple hierarchies, for example, drug hierarchies curated based on different criteria, one could be based on drug chemical structure similarity, and another could be based on drug side effect similarity. How shall we integrate them into one hierarchy or one class embedding space? The key idea is to decompose each hierarchy as we did before and to let node embeddings be shared across different hierarchies. So here, xi represents the embedding of node I by integrating different hierarchies. Now our method can be used to integrate information from accumulating and diverse biological hierarchies. So what should we do if the hierarchy has multiple node types and edge types? For example, a hierarchy could have not only drugs, but also disease and genes. How should we embed these nodes? We introduce additional parameters to model different edge types. Because the hierarchy could be very large when it has multiple node types and edge types, we sample edges according to the frequency of different path types. So in each iteration, we first sample a path type. Then we sample a specific path according to this path type. This method enables us to incorporate data set from other sources into biological hierarchy, and then to jointly analyze all of them. For example, we have incorporated electronic medical records into this kind of biological hierarchy and jointly analyze those hierarchy information, electronic medical records information, and the clinical notes information. So what should we do if we don't have the class hierarchy? This time, we need to build a new hierarchy. We jointly mine scientific literature and biological database. We first build a gene class hierarchy for the specific problem we want to study using biological dataset. For example, here we build a cancer hierarchy to model tumor evolution. Each node here is a cancer gene set by clustering patients' genomics data. We then mine biomedical phrases from literature and align each gene cluster with one or more phrases. In this way, we can have a new hierarchy for follow-up predictions. And this method enables us to update, correct, and build biological hierarchies. And then our method can be broadly applied to other biomedical applications. So this is the first part of this talk. We use class hierarchy to generate class embeddings for never before seen classes. Our representation method has two steps. First, we use equilibrium distribution to capture the global structure similarity. Second, we reduce dimensionality of those random walk equilibrium distribution. Next, I'm going to talk about the second part, how we use these class embeddings to classify samples into a never before seen class. So this is the problem setting for the never before seen classification problem. The first input is training samples. The second input is class embedding we got from the representation step. The goal is to classify test samples, and many of them might belong to a never before seen class, like the green ones or the purple ones here. So during the training stage, we want to find a transformation to project samples from the sample embedding space to class embedding space. The optimization goal is to map training samples as close as possible to their classes in the class embedding space. For example, in this case, you can see that the optimal transformation is like rotate a little bit and squeeze in this dimension so that orange samples are close to the orange class, pink samples are close to the pink class, and blue samples are close to the blue class. During the test stage, we use the same transformation to project test samples. And test samples will be classified into the nearest class in the class embedding space. For example, those ones will be classified into the orange classes because it is in the orange region. The key of our method are those samples in the green region. They are classified into the green classes, and we have never seen any green samples before. So by using those class embedding, now we enable the classification of samples into a new class. So let's look at the implementation. During the training stage, we want to find the W's and minimize the cross-entropy loss. During the test stage, we use the same transformation to classify each sample into the closest class. So generalized zero-shot learning could be easily overfitted and biased to scene classes. In the extreme case, all test samples might be classified into scene classes. So we often use a linear transformation in the training stage to avoid overfitting. We also did a calibration after the test stage to normalize prediction scores between scene classes and never before seen classes. Our method has been applied to a few important biomedical applications, such as protein function prediction, single-cell prediction, and patient disease prediction. Today, I'm going to talk about two of these applications. First, let me talk about the single-cell prediction. What is a single cell? If you think our human body as a city, okay, a city on the map, then each cell is a building in this city. The cell type is the building type, like restaurant or school. The goal is to classify cells into cell types, and we have only seen 5% of the cell types in the training data. Like we only know 5% of the building type in this map, and we want to use those topological information to infer the building type of other buildings in this map. This is one of the most important problem in biomedicine right now, because we detect diseases by somehow counting different types of cells in our body. So this is a complete blood count test, which is often the first test we get if we feel sick. In these tables, we can see that 3 of those rows are the counts of different cell types, white blood cell, red blood cells. And 5 other rows are different properties of cells. So from this widely used CBC test, you can see how single cell can be used to detect diseases. In 2018, there's a very exciting technology breakthrough called single cell sequencing technology. And with this technology, we can now measure the features for individual cells. And those features are like gene activity score. And in biology, we call them gene expression. And single cell technology let us clearly see the feature of every single cell, like here in a fruit bowl, rather than seeing the averaging features of many cells in a big organ, like here in a smoothie. If we don't study these cells individually, we will not be able to tell which part of the organ is sick, or we might not even know that it is sick. Because now we can see every individual cells, we can see the features of every individual cells. We will then see lots of never before seen cell types. Then predicting such cell types is an emerging problem. This is the experimental setting of our method. There are 2 inputs. The first is the gene activity feature scores of 500,000 cells. Each is a 20,000 long numerical vector. The second is the hierarchy of cell types. It is an expert curated hierarchy. It has more than 2,000 cell types, but only 98 of them are seen in the training data. So we now want to use our method to classify those cells. First, let's look at the cell type prediction performance. X-axis is how many new cell types are there in the test data. A larger x-axis means that there are more never before seen cell types. And the pink one is our method. We can see that our method outperforms all the comparison approaches. And even when 80% of the test data is from a never before seen class, our AURC is still greater than 0.7. The key observation is that the improvement of our method is larger with the increase of never before seen classes. By embedding cell type hierarchy, we enable the classification of cells into new classes. And this kind of never before seen classification often cannot be achieved by human experts because they don't have the class attributes. We then studied the generalization ability of our method. Generalization ability is very important in biomedicine when we want to integrate and jointly analyze multiple data sets. Here we collect 27 data sets from different species of tissues. We train on one data set and then predict the cell types on the remaining 26 data sets. We can see that our method has a strong generalization ability. For those cell types we saw in the training data, the AOROC is greater than 0.95. Even for these 7 cell types that have never been seen in the training data, we still have an AOROC greater than 0.8 for 4 of them. Because we can accurately classify cells into cell types now, we can also then do some post-processing to find data-driven class attributes. And those novel class attributes are exactly the knowledge that human experts want to know. We observe very good performance here, and our method, the red one, is comparable to expert-curated attributes in classifying new cells. So this is the first application, single-cell classification. Now let's look at the second application, how to predict protein functions. Human body consists of trillions of single cells, and each cell consists of millions of proteins. If you think single cells are building, then proteins are like windows, carpets, or bricks in the building. And the protein function will be the property of these bricks, windows, and carpets. For example, is it waterproof, soundproof, and fireproof? The goal is to predict the function of each protein. The challenge here is that most of the functions have very few training samples. For example, in human, about 15% of functions has less than 10 training proteins. So this is now a few-shot learning problem. Why is protein important? From this CBC result, you might notice that 8 of them are counting or related to cells. And the last one is actually an indicator of protein. So we also need to monitor proteins to detect human diseases. Let's see the experimental setting. There are 2 inputs. The first is the feature representation proteins. The second input is the hierarchy of functions. We want to use our method to do the classification here. Let me first introduce how to represent proteins from different species. To get the feature embedding for each protein, we integrate proteins from different species. There are 2 types of edges. One is edge connecting proteins across different species based on sequence similarity. The other is edge connect proteins within the same species using molecular networks. So here, the networks could be constructed either by biological experiment or by mining scientific literature. This kind of large-scale network enable us to embed proteins from different species into the same low-dimensional space, so that we can transfer biomedical experimental result like drug effectiveness from other species to human. This process also creates a very unique data set, including 60,000 nodes and more than 200,000 labels. Here are the results of our prediction model. X-axis represents classes in different categories based on how many training samples are there for each class. The blue one is our method. We can see a significant improvement in comparison to existing approaches, especially for those functions with very few training samples. The improvement of our method comes from using the hierarchy to classify proteins into functions with very few training samples. This is how we use class hierarchy to classify samples into never-before-seen classes in biomedicine. First, we can now better apply zero-shot learning to biomedicine using class embeddings generated from class hierarchy. For biologists, they can now make predictions in tasks with no training samples, like new drugs, new disease, and new genes. Okay, so those are the first 2 parts, representation and classification. Now, The next question is, how can we interpret these predictions? We use scientific literature to help us find those interpretations. So how to interpret the prediction of a never-before-seen class? Many existing methods, many interpretable methods, find important features within this data set. For example, here, glioblastoma is one of the most aggressive cancer with average survival time of only 3 months. From this glioblastoma genomics patient data set, we can find which genes are interpretable for cancer survival. And by using some existing interpretable method, we might find that these 7 genes are interpretable. But is this information enough? So this information is very helpful, but we might want to know a bit more about the mechanism. That is, why these genes are interpretable. Can we interpret these interpretations? This kind of question is actually the key question in biology. They are the new knowledge biologists want to know, especially when trying to interpret something we have never seen before. So how should we answer this question? Let's look at this character string. So these characters can interpret, or these characters can interpret my talk, and they are the most representative information of my talk. But can you understand this string? You don't even know how to read these strings because the structure information is missing here. There's no space and no punctuation in this string. In biology, it's the same. Even if we found out that these 7 genes are important, they are interpretable for cancer survival, we still don't know why they cause glioblastoma. In fact, many biologists spend their whole life just studying 1 or 2 single genes. So it will be very difficult for a single scientist to know why these 7 genes together cause glioblastoma.
Sheng, can I interrupt for 1 second? Sorry to.
Yeah.
Can you just explain a little bit more what you mean about interpretability and if these 7 genes are, what do you mean here?
Okay, so here I train a supervised learning model using the genomics feature of those genes to predict cancer patient survival. I train a supervised learning model, maybe a deep learning model, a tree-based model. Then I call in external explainable method like tree explainer, sharp, lime, or those kind of explainable method. Those methods tell me that those 7 genes is the interpretable features. They have the highest feature values, or they are most interpretable for the prediction result for a supervised learning method.
Thanks.
Okay, yeah. So an external interpretable method tells us that those 7 genes are important, and then we want to know why are they important. Okay. So this is the problem. So to understand a character string, we need to know the structure at different levels. Like we need to know the word, clause, sentence, and the paragraph. We cannot understand a document by only reading a character string. In biology, it is the same. To understand why these genes are important or interpretable in this disease, we need to know the information between genes and diseases. In biology, we call them scales, different scales of information. Our biomedical world transmits from nanometer scale genes to micrometer scale cells to millimeter scale tissues, and finally to the disease we observe on the centimeter scale organism. So now the challenge is that we only observe gene scale information and the large scale disease information on our human body. How can we identify the scales between them? It is like you are reading a book, and this book is only written in characters without any space and any punctuation. How could we understand this book? So going back to this example, some of you might be able to understand this character string without any punctuation or space. This is actually the title of my talk, Learning for Never Before Seen Biomedicine. So if you can understand this sentence without punctuation, you are actually using some multiscale knowledge graph you learned by reading other texts or other documents. You infer that learning shall be a word and never before seen shall be a high-level structure, then this is a combined phrase. So the same in biology. we need such external knowledge graph to help us group these genes into gene cluster or gene modules, then to cells, then to tissues, and finally we can understand why they cause this disease. But we don't have this kind of multi-skill knowledge graph in biomedicine, and what should we do? So we built a multi-skill knowledge graph from literature. It integrates millions of scientific papers and biological database records. There are many other knowledge graphs. The main novelty of our knowledge graph is this multi-skill structure. We have a very unique node type called multi-skill gene modules. So each gene module is a set of genes that work together in the biological system. And these multi-skill gene modules connect genes to disease at different scales. We mined 27,000 novel gene modules from literature, so our knowledge graph can be updated all the time with new literature. By using this knowledge graph, we can now understand why these 7 genes are interpretable, or why are they important or predictive for this glioblastoma cancer. So let's see this example again. Here, an external interpretable model tells us that these 7 genes are interpretable, but we don't know why. Then we can answer this question by searching these genes and the glioblastoma in our multi-skill knowledge graph, and then extract the subgraph. We can now, for example, PCSK2 is important because it affects microglial cell proliferation, then EGFR pathway, and then finally cause glioblastoma. Each edge here is linked to an external evidence. It could either be a biomedical database record or a scientific paper abstract which co-mentions the 2 nodes sharing this edge. We evaluate the performance of our method using this knowledge graph on 7 biomedical prediction tasks, like predicting the function given a set of genes or predicting the associated genes given a disease. We found that integrating biomedical database and scientific literature can do much better on these 7 tasks than only using scientific literature. We also build a web server so that user can query any number of genes, gene modules, drug, and disease here, and they don't need to use our GitHub repo or code, and they can use the web server to support their research. After querying different gene, module, drug, or disease, we will return a multi-skill evidence subgraph for the user. We also have done some other work in text mining to support information extraction from noisy text. For example, in this online health forum post, the user mentions multiple side effects from different drugs. So side effects from different drugs are mixed together. We built a probabilistic topic model to figure out which side effect is associated with which drug. And this model can be used to model lots of text data jointly, and we up this model using the EM algorithm. Our method has been used by doctors to analyze 300,000 patient records from 6 hospitals. So this is the last part of this talk. I introduced a multi-skill knowledge graph that can help us better understand the mechanism in biomedicine discovery. For biologists, they can now obtain up-to-date and multi-skill evidence to further interpret their discovery. Let's summarize my talk and the previous research. So using our representation method, we can now provide personalized treatment for 6,000 patients. Our method has been used by Mayo Clinic and NIH Knowing Center to identify drug mechanisms. We also introduced a new biomedical data set for machine learning researcher to study zero-shot learning in biomedicine. In the classification part, we talk about 2 important problems: single-cell prediction and protein function prediction. Our method has been used by the Chen Zuckerberg BioHub to predict cell types in one of the largest data sets. In the interpretation part, we introduced a multi-skill knowledge graph. Our knowledge graph has been used by 200 scientists in NIH NCAT, and another text mining method has been used by doctors to analyze electronic medical records and clinical notes. And these three steps together build up a comprehensive framework and let us study biomedicine from no labels to new knowledge. The most exciting thing is that this is a general computational solution to many different biomedical problems, like cancer survival analysis, single-cell prediction, protein function prediction, and drug side effect prediction. Next, I'm going to talk about a few future works in representation, classification, and interpretation. The first problem in representation we want to explore is how to do high-order zero-shot learning. High-order prediction means that predicting the effect of combination. For example, predicting synergistic effect of drug combination in cancer treatment. This problem could be very challenging because we only have training data for low order for the single drug, and we don't have training data for high order multiple drugs. I think a key solution here is to build better representation learning method that can represent k items jointly. So how to represent one drug, two drug, and three drug combo so that they are comparable to each other. The second future work is to do transfer learning across population during classification. Most of the bio data set are collected from certain population. For example, UK Biobank is one of the most widely used data set. However, more than 19% of patients there are Europeans. So machine learning model trained on this kind of data set may not be generalized well to other groups. How can we develop effective transfer learning algorithm so that we can generalize our machine learning models? The third future direction is a more broad one. How can we better use NLP in biomedicine? Right now, most of the NLP algorithms in biology focus on analyzing text data in biomedicine, like scientific papers and clinical notes. I think it will be very exciting to integrate biological data into the NLP model. So we not only analyze biotext data, but we also integrate genomics data, mutation data into the NLP model. For example, can we generate sentences using abstractive summarization from literature and using the genomics data to describe disease mechanisms? And how can we integrate bio data and text data? Because bio data often has more coverage of genes, but it often has cross-screen relations. On the other hand, text data like scientific literature has less coverage of individual genes, but often has more detailed, more fine-grained relations. How can we align and integrate those two different data sources? So this is all I want to talk today. Thank you for attending to my talk. I talk about how to represent, classify, and interpret never before seen biomedicine. And by doing this, we can build a general solution that enable us to transfer from no label to new knowledge. I would like to thank all of my collaborators, and I'm very happy to take questions. Thank you for attending my talk.
Thanks so much. Really exciting and very, very clear talk. We have time for a few questions.
Mm-hmm.
I will go first. I actually have so many questions. I'm glad we have a one-on-one later on. But picking up on your future exactly in that slide, what kinds of descriptions do you think we should make, and how should we communicate to scientists the uncertainty around the disease mechanisms?
Mm-hmm.
A lot of our generative models, hallucinate. So there's two forms of uncertainty: whether the generative model is actually accurately reflecting what is found in the papers, but also whether the papers are clear about what the mechanism actually is. So how would you communicate that to the user?
Yeah, I think one interesting mechanism to generate is to generate the mechanism of a gene set. So I think diseases are interesting, but what is more interesting is a novel gene set, because most of the time we can represent a disease or a new disease as a set of genes. So you can, so given a set of genes, like 10 genes or 20 genes, and we know the individual function, individual sentence for each individual genes, how can we summarize them? Summarize those 10 genes. What's the function, what's the mechanism of those 10 genes work together? This is a very large search space. We have 20, human has in total 20,000 genes. And most of the time biologists, they find 10 or 20 genes they are interested in. And then how can we generate a sentence to summarize them? We have some ground truths to validate some biologist written sentences for about 30,000 gene sets. So we have a very good data set to do validation. And those are not from literature, those are from the gene ontology database. So people write the detailed sentences for each function, and each function is associated with like 10 genes or 20 genes. So this will be, I think, something that is very interesting to generate.
Can I, I don't know, Dan, if you want to keep going. Thanks for the talk, Sheng. I'm just wondering whether you can comment. I feel like a lot of these computational methods, especially for this kind of mechanism discovery type of task, the goal is really to help scientists, help biologists prioritize what experiments they do to then verify some of these mechanisms. So can you talk about like how, maybe how to integrate the literature and these knowledge bases better to help prioritize the hypotheses that you're essentially generating?
I think most of the time it may not be a prioritized problem. So most of the time it's like they find some gene set from their experiment, and then they're trying to know the mechanism, and they don't have any hypothesis in their mind, and they just want to expand those mechanisms. And then they may want to search the literature to see what individual genes, to see what individual gene does. And then what we can do is actually trying to accelerate this process to help them automatically read the literature. This is what I think. If we want to help them if we want to help them prioritize their mechanisms, I think we also need to further incorporate, we need to directly incorporate their biological data set, like their genomics data or their mutation data into our machine learning model, so that we can better help them prioritize their experiment. And this is why I think it's very interesting, very important to integrate biological data set into the NLP model. For example, you can use biological data set as a prior knowledge, or as a build a pre-trained model on genomics data, and use those pre-trained model to do some scientific literature analysis, using those genomic-based pre-trained model to do relation extraction, to do something in scientific literature.
Yeah, that makes sense. But I guess for any, any gene set that comes out of these enrichment analyses, there could be multiple explanations as to how those genes work together to cause disease. And I guess like, how do you represent that kind of uncertainty?
Yeah. So this is like in this paper, so we have a set of genes, we have a disease, then we have different paths here to reach this disease from these genes. Maybe it can go from here, maybe it can go from here. And then you will find that those different paths is related to different other diseases. So some may relate to like a blood vessel, some may relate to some cell differentiation. And this is why I think we should also consider the specific domain that people generate this data set, that people find those genes, whether it's from some kind of bioassay, some cell developed assay, they generate those gene set, or from some other like patient with some blood disease, they generate those gene set. So I think we need to take into account those kind of specific biodomain so that we can better know which path or which hypothesis generate this disease.
Thanks.
Other questions?
I have another one if there's a chance. I think you mentioned on one of your latter slides that the data that's been collected and generated is not really representative of like all populations. So some of the relationships that are mined from the text and are more supported by textual evidence, it might be more representative of common diseases in these like well-studied populations. So what are some things that can be done, like when you're talking about these never before seen diseases or mechanisms or populations, what are ways to kind of offset this distribution problem?
Yeah, I think so. This is what I mean here, trying to integrate or align biodata and text data. So from text data, for scientific literature, we can get some maybe very general gene-gene relationship, and this could be applied to many different populations. We didn't specify the population in text data, so this might be a case. And then we can find a data set, like population-specific biological data set, that we can try to align those data sets. We use the population-specific biological data set, population-specific genomics data to help us filter those general relationships from text data. And this is how we can try to find population-specific relationships from text data. So again, we can use population-specific genomics data to help us do some filtering to do it.